everyone. So today we're going to look at the nervous system, in particular the synapses and how action potentials are transmitted across the synapse to the neurons. So so we know that the basic unit of the nervous system is a neuron. A neuron is a nerve cell, basically. So in the body, nerve cells connect with one another. So neurons would link different cells, and that these neurons are able to conduct impulses between them. So neurons vary in size and shape, but we'll be looking at something very, very specific. It's the synapse. So we know action potentials are generated and they are propagated along the neuron until they reach the end of the neuron, so the end of the axon. Now, as you can see from this picture, this is a motor neuron and it's ending in knobs. So these knobs here at the terminal end of the axon is called synaptic knobs or synaptic bulbs. So the thing is, let's say an action potential is coming this way, which it would be because axons take the impulse away from the cell body, so away from the cell body. So you have an action potential coming here, if you follow my cursor, until it arrives at the end. Now this end would most probably be connected to either an effector in the case of a motor neuron here, or it could be another nerve cell. So if we look at this picture here, we can see synaptic knobs. They're at the end of the axon, and they are making like a little connection to the postsynaptic neuron here. So there will be terms used like presynaptic neuron, which has the nervous impulse coming to the end, to the synaptic knobs, and the next neuron, which we want to transfer the nervous impulse to, is called the postsynaptic neuron. And the whole thing here is called a synapse. So as you can see from this picture, there is a little gap here between the presynaptic neuron, the postsynaptic neuron. So yes, membranes are of the different neurons are not joined, they're not connected, so there is a little cleft. It's a very, very small cleft. It's just 20 nanometer in diameter. Yeah, okay. So having said that, the nervous impulse, when it arrives here at the end of one neuron, it can't just jump at this synapse to the other connecting neuron. So how can the impulse be transmitted? It's by the action of chemicals called neurotransmitters, which are released at one end, more precisely from the presynaptic neuron, at the end of the axon, to the postsynaptic neuron. Okay, so we are going to look at this in more detail. So here we go. Here's a little picture showing what happens to the action potential as it arrives to the end of the axon, which ends in the synaptic knob, as we mentioned. So we'll just go through it stepwise. So we can see from the green arrows in the left diagram, over here, we can see the action potential arriving. So when an impulse arrives at this end, called the synaptic bulb, what it does to the membrane is it opens calcium channels. These are voltage gated. So when the action potential arrives here, because of depolarization of the membrane, this calcium ions open. Hence the term permeability to calcium ions. So obviously calcium ions diffuse from the outside into the synaptic knob. And these calcium ions do something in particular. So what they do is they stimulate the movement of synaptic vesicles. So in the synaptic knob are some vesicles in which there are neurotransmitter substances. A few examples of neurotransmitters in the human body would be acetylcholine, noradrenaline, dopamine, glutamic acid in the brain, dopamine in the brain. So these are a few examples, neurotransmitters. So we were saying that these calcium ions, what they do is they stimulate these vesicles which contain those neurotransmitter a particular neurotransmitter substance. They stimulate the movement of these vesicles towards the membrane, the presynaptic membrane. So you can see these vesicles arrive here. And what happens is these vesicles, by the process of exocytosis, just bulk exchange, fuses with the presynaptic membrane. So the vesicle fuses here with the presynaptic membrane and releases the neurotransmitter into this cleft, the synaptic cleft. So you can see what happens to the neurotransmitter. Once it's released, it diffuses down the cleft and it binds to receptors on the membrane of the postsynaptic neuron. So obviously, you do remember that receptors are they are very specific to a particular neurotransmitter. So the neurotransmitter binds to this receptor. So when that happens, what goes on next is once this neurotransmitter is bound there, then the membrane, post-synaptic membrane, becomes depolarized. So what that means is we know that the inside of neurons, which are resting, is negative and the outside is positive. So depolarization means that the inside becomes less and less negative, more and more positive, because sodium ions move, sodium channels open and the sodium ions move into the cell. So the inside of the cell would become less and less negative, more and more positive because of the sodium ions. So that's what a depolarization is. So the postsynaptic membrane becomes depolarized and an EPSP is generated. So EPSP basically means excitatory postsynaptic potential. And something important to note is that the EPSP will depend, so the intensity of the EPSP and the extent to which the postsynaptic membrane becomes depolarized will obviously depend on the amount of this neurotransmitter that is released into the cleft. The more neurotransmitter released, the more receptors are filled, and the higher the degree of the depolarization. And so this depolarization is called an EPSP. And when the EPSP reaches threshold intensity for an action potential to be generated, then this creates an action potential in the postsynaptic membrane. So you can see your action potential, it arrives and the new vesicles, neurotransmitters release, and the action potential is then generated in the post synaptic membrane here. And it's then propagated from this point onwards. It's propagated along the whole length of the postsynaptic neuron. Now, how about the neurotransmitter? So let's see. So neurotransmitters, once they're bound to the receptor, imagine if they continued staying bound to the receptor, that would mean continuously action potentials being generated here in the postsynaptic neuron. So that means nerve impulses would just continue being fired off in the postsynaptic neuron. And that's not something we want, because that would result in maybe, if the neuron's connected to muscle, that'll be spasms. There would also be something called fatigue of the synapse, where eventually the neurotransmitter here, it just can't keep up with so neurotransmitters actually get exhausted. So if action potentials are continuously coming, neurotransmitter is being released, and this here, it gets really the transmitter substances get exhausted and the synapses fatigue. So at some point, the nervous impulses would not be continuously transmitted here to the postsynaptic neuron. So coming back, so neurotransmitters, therefore, then need to get hydrolyzed into breakdown products. So they do get hydrolyzed. So there's enzymes here that break down the neurotransmitters, which are bound to the receptors, into the products. So if we take the example of cholinergic synapses, where the neurotransmitter involved is acetylcholine, then acetylcholine would be having an enzyme called acetylcholinesterase that would hydrolyze the acetylcholine into choline and ethanoic acid. So once these products are there, acetylcholine has been hydrolyzed, then the breakdown products diffuse right across the synaptic cleft to the presynaptic membrane. So the breakdown products diffuse across the cleft and they're reabsorbed into the synaptic bulb. And what happens to these two products? They are then resynthesized into acetylcholine. So, so this is it's very good for the cell to resynthesize, right? It's less wastage of resources. So let me just point out that this resynthesis of the neurotransmitter involves ATP from mitochondria. Just have a very, very pretty picture here. This is there's actually a scanning electron micrograph of the end of the axon, so a synaptic knob that's been cut off here. So you can clearly see the orange, the yellow, and the blue. These are actually, as you can guess, these are vesicles, neurotransmitter vesicles. So synapses. Let's see. So what organelles what do you expect to find in abundance then at the synapse? So obviously you would expect to see a lot of mitochondria, and the mitochondria will be more abundant here in the presynaptic neuron. So mitochondria, as we just said, would synthesize ATP, which is required for resynthesis of neurotransmitter. They would expect to see a lot of vesicles. Remember, vesicles, how they formed? They formed from the Golgi apparatus, or they formed from the rough ER. So rough ER is an organelle, is a membrane system of tubes and sacs. So the rough ER will be a membrane system of sacs, and they have lots of they have lots of ribosomes on top of them, and the ribosomes will synthesize proteins, and the proteins would then be packaged into RER vesicles, shipped to the Golgi apparatus here, which would then add some tails, some carbohydrate tails to the protein. If you remember, and these vesicles then bud off. So protein is in there. Yeah, so see lots of these vesicles for packaging the neurotransmitters after they're synthesized. Yes, okay, so that's in short. So synapses, then we may be asking ourselves questions like, what's the point of having a synapse? Well, we just mentioned that actually synapses have a lot of advantages. So we may argue that synapses slow transmission because of the gap, but actually this is very, very small gap, and it only takes a fraction of a second, of a millisecond, for the diffusion of these acetylcholine across the cleft. So that's not a problem. There are more advantages to having synapses. So synapses ensure that transmission of the nervous impulse is in one direction only. So if we're looking at the neuron here, we're thinking nervous impulse coming that way. So and ensure that the action potential is going off in this direction to the next postsynaptic neuron, because synaptic knobs are only on one end of the neuron, and also the neurotransmitters would be released only from the presynaptic neuron. So there's no way that the impulse, once it comes here, goes backwards. So it ensures that action potentials are going in one direction. So unidirectional transmission of the synapse. Also, the synapses, they protect muscles and glands from being overstimulated. So as we just mentioned, if there's too many impulses in a short period of time, so this continuous transmission of action potentials here, it exhausts the supply of neurotransmitter at the synapse, which means if, let's say, this neuron is connected to a muscle, this protects the muscle from fatigue. So the synapses fatigue actually because the neurotransmitters, they can't be released anymore. We've used up all the neurotransmitters. So this protects muscles from overstimulation. Something else about synapses is oftentimes the activity of lots of different synapses is integrated with a single postsynaptic neuron. So this provides flexibility and allows changing responses, allows a variety of responses, more complex responses to a particular stimulus. So that's those are a few advantages of having synapses. Here's a lovely TEM, and we can see clearly in this transmission electron micrograph, we can see these neurotransmitters vesicles very, very clearly. And we can see there are none on this side of the membrane, the postsynaptic membrane, but there are loads of these vesicles on the presynaptic side of the synapse. So that was just a little bit about synapses. If you have liked this video, please don't forget to put a like and subscribe to my channel, which is Biology Bits for you. And if you need some help with your biology, if you're struggling, if you have any, or you'd like to prepare for the exams, don't hesitate to get in touch. Thank you very much and have a nice day. Bye.